Hello. The topic of our presentation is regulation of gene expression via the core promoter and the basal transcriptional machinery. My name is Talani Sileodu and my partners are Damilola Balogun and Edna Rodriguez.
In this review, we will discuss some features of RNA polymerase II code promoter and provide some example of role of code promoter and basal transcription machinery in the regulation of gene transcription. The code promoter is the region of DNA that directs the accurate initiation of transcription by RNA polymerase II. In the fork transcription, there is either a single measure transcription, the start side, or several start sides within narrow region or several nucleotides. Focus transcription is the predominant mode of transcription in simple organisms. In the dispersed transcription, there are several weak transcription start side over the broad region of about 50 to 100 nucleotides. Dispersed transcription is the most common mode of transcription in the vertebrate. For instance, dispersed transcription is observed in about 2/3 of human genes. In the vertebrate, Fox transcription tends to associate with the regulation promoter, where dispersed transcription is typically observed in constitutive promoter in CPG is lined.
The analysis of focus co-promoters has led to the discovery of sequence motifs such as the Tata box, BRE, MCE, and DPE. Focus transcription typically initiates on the A nucleotide on the initiator and is usually designed as a +1 position. RNA polymerase can synthesize RNA from DNA, but it's not usually able to recognize the core promoter. This requires additional factors known as the basal transcription factors. Some of these factors are able to mediate transcription from the Tata-driven promoter while repressing the DPE-driven promoter, while others do the opposite. Examples of some of these basal transcription factors include TF2A, TF2B, TF2D, TF2E, TF2F, TF2H, and NC2. The Tata box is the best core promoter and is located at -31 to +30 on the core promoter. The DPE is usually found downstream of the initiator, located from +28 to +33. The BRE usually works in conjunction with the Tata box, while the MCE works in conjunction with the DPE sequence motif. The Drosophila hoax gene was analyzed, and this has revealed new insights into the role of the core promoter in a regulatory network. Caudal is a sequence-specific DNA binding transcription factor, and it is a key regulator of the hoax gene. This diagram basically explains how caudal mediates transcription in a core promoter containing either DPE or Tata box sequence motif. The first one basically shows that caudal works in conjunction with the DPE core promoter motif and is able to mediate transcription. The second picture shows that caudal is also able to mediate transcription in a DPE core promoter, even in the presence of BREU, which means the presence of BREU doesn't affect the work of caudal. The third one shows that the mediation by caudal on the Tata box sequence motif is not as effective as it would be in a DPE core promoter motif. The fourth diagram shows that the presence of BREU in a Tata box core promoter motif further suppresses the action on caudal, meaning it doesn't lead to the expression of the gene on that core promoter motif. This slide is talking about how DPE and Tata specific activators work. Transcription factors bind to enhancers, but only activate transcription from promoters with the appropriate core promoter elements. The core promoter containing both the Tata and the DPE motifs can be activated by either DPE or TATA-specific activators. Transcription levels can be further regulated by the presence of the BREU as well as other core promoter motifs. This basically means that if a Tata box core promoter is present, then only a Tata-specific activator will be able to activate the whole process of transcription. But in a situation where there is the presence of the DPE and both the Tata, either the DPE specific activator or Tata specific activator can work in conjunction with the core promoter motif to continue the process of transcription. Over the past slides, we have been talking about how to regulate gene expression using the core promoter, but that is not the only way genes can be regulated. It is also possible to use the TRF in transcriptional regulation. There are three types of TRFs that have been identified. There's the TRF1, the TRF2, and the TRF3. The TRF1 does not exist in yeast and humans, but it is present in Drosophila, and it usually participates in transcription by RNA polymerases 1, 2, and 3. The TRF2 is present in most eukaryotes and is involved in transcription by RNA polymerase 2, and it does not bind to Tata box sequences and cannot replace Tata binding proteins in vitro. The TRF3 appears to be present in only vertebrates, and it is the TRF that is most closely related to Tata binding proteins, and it can also bind to Tata box, thereby supporting Tata dependent transcription.
The core promoter and basal transcriptional machinery are two important yet relatively unexplored dimensions in the regulation of gene expression. For instance, transcriptional enhancers would ideally be studied in conjunction with their cognate core promoters. Alternatively, a new generation of reporter vectors could be designed with core promoters that function with both Tata and DPE specific enhancers. Therefore, in the future, it will be essential to consider and to incorporate these factors in the analysis of gene regulation. The increased appreciation and understanding of core promoter motifs and basal transcriptional factors will lead to new and exciting discoveries and ultimately provide a more complete and accurate view of biological regulation.